All right, thanks for joining me. I'm really glad to be here, even though it's just digital. I wish we could talk in person, but this is the next best thing. Today, I'm talking about your stem cells, upgrading S.A.D. stem cells. What's S.A.D? Standard American Diet. A uh, well-designed acronym. I have no conflicts of interest. I have no disclosures here. I actually do DNA consulting through my AJ Consulting company, but it's not really a conflict of interest. So Juan Murillo, in 2008, he's a National Geographic pilot. I crashed his plane and broke his spine. Neurologists from Florida and Costa Rica, actually, they told him, we've seen a ton of spinal injuries like this. Nobody walks again when you have this type of injury. And he couldn't move his legs. I mean, literally for weeks. He saw a specialist, like I said, in Miami. They all said the same thing, you're paralyzed for life. And then after one stem cell infusion, his pain went from a 10, right, down to a 3. And 4 months later, the second stem cell infusion restored some feeling in his leg. And then he started moving his toes. I mean, he just got better and better. 4 stem cell injections later, he's walking. He's even jogging now, to be honest. And he's got a little bit of numbness in his leg, but I mean, pretty phenomenal, considering all these doctors said, yeah, you're not going to walk ever again. A different person also named Juan, 2009, he fell down a riverbank, broke his neck, broke his back, completely paralyzed. After 8 months, he regained some use of his arms, could wiggle his toes, etc. Stem cell injections, again, eliminated his pain. And he's now independent. He lives on his own. Similar story. I mean, just phenomenal results with these stem cell injections. So Neil Reardon is where I'm getting these stories from. He's down in Panama. He's treated over 100 patients with spinal cord injuries. 70% have experienced restorative function. Now, I'm not going to just talk about stem cell injections today. I'm going to talk partially about injecting stem cells for maybe the first half of the talk. But then the second half, I'm going to actually tell you how to upgrade your own stem cells. And it's important to have both of those components because, you know, there's a time and place for both of these things. Obviously, everybody should be upgrading their own stem cells, but it helps to understand why inject stem cells. Why are people doing this? What are some of the benefits? In order to even understand how to upgrade your own. And I do stem cell research at the Mayo Clinic personally, so I have some familiarity with injecting stem cells. But first, what are stem cells, right? Stem cells are less complex cells. Number one, they can divide indefinitely, so they can make more stem cells, but then they can also produce more specialized cells like you see here. So you can produce brain cells, you can produce heart cells, you can produce kidney cells from stem cells. And of course, all your organs actually have stem cells in there. And so they can keep making more stem cells or repopulate those organs that you need repopulated. There's 2 major types of stem cells. There's embryonic and adult stem cells. Embryonic cells, they develop 3 different ways into the ectoderm, the mesoderm, and the endoderm. So of course, when egg and sperm cells, they only have 1 copy of DNA. When they fuse together, that's where you get the 2 copies of DNA that every cell in your body has. You get the copy from your mother, the copy from your father. Those cells start dividing, and that's where you get this, these stem cells separate and ultimately form you, right? So the embryonic stem cells, in 2012, there was a scientist named Shinya Yamanaka, Nobel Prize winner. He discovered 4 genes. They call them the Yamanaka factors. And basically, if you take out stem cell or skin cells or liver cells or any kind of cell that's an adult cell, and you put these Yamanaka factors, you insert these genes, these 4 genes into those cells, that adult cell reverts back to a stem cell. So obviously a Nobel Prize winning finding. The problem here is that embryonic stem cells are known for dividing super rapid, but also becoming cancer. So you don't want to take, it's nice that you can take your own stem cells out of your skin, revert them back to embryonic cells, but again, cancer risk. So there's literally no successful clinical trials using embryonic stem cells. And they're also antigenic, which means it triggers your immune system. So certainly you wouldn't want to take adult cells from somebody else, revert them back to embryonic, put them into somebody else, or put them in yourself. You'd have a real tough time getting them into your body. Your immune system would react to them. You'd get inflammation, potentially cancer. So it's a lose-lose. Adult stem cells, on the other hand, right? That's embryonic. This is adult. You can isolate those and put those back in your body, and you don't have those issues. So there's 4 commonly pooled sources from adult stem cells that we use. Bone marrow is one of them, fat, blood, and umbilical cords. With bone marrow, fat, and blood stem cells, you can remove them from your own body. Like I say, you can re-inject them. They call that autologous stem cell injections. Autologous, auto, it's yourself. And here's some of the conditions that are literally being treated with clinical trials using some of these stem cells, these autologous adult stem cells. I highlighted osteoarthritis, cartilage repair, and tendon injuries because, I actively work on some of these areas. But there's a ton of areas here, and you can see a lot of potential. The thing about the autologous stem cells, obviously with all these different sources, choosing the correct source is complicated. My preference, like I say here, is actually these fat cells, because everybody's happy to get rid of fat, and blood has so few stem cells. Bone marrow has some stem cells, but it's really painful. You got to do quite the procedure to get into there. The problem with fat, as you can imagine, what are Americans eating, right? Lots of McDonald's, lots of processed food, lots of garbage. Basically, some healthy people have amazing fat. Some people have terrible fat. There's a wide range. I call it McDonald's fat, right? I call it McDonald's stem cells, because basically we have stem cells in our fat, and those stem cells, if you're constantly exposing them all these problematic chemicals, all these oxidized seed oils and things like that, basically those stem cells are all messed up. And an issue in science, right, is a lot of scientists just kind of lump it all together. They say, well, we got fat cells from this person and it didn't work, right? We got fat cells from this person, it did work. When we used the stem cells, we re-injected them, things like that. So they're just lumping it all together as if it's one thing. But there's a nuance here. Now we're finally, acknowledging this. Scientists literally used to laugh at people when they talked about how there's, your fat stores toxins and all this. They always get triggered by the word toxin. But in reality, now they're publishing papers. This is literally a title from a paper: persistent organic pollutants in adipose tissue should be considered in obesity research. And by the way, I have all the references. If you need the references, I'll give a slide at the very end. You can find my consulting website. You can send me an email. I'll shoot them to you. But the point is, we're finally acknowledging that these toxins that we're being exposed to are literally storing in your fat and disrupting these stem cells. So that's a problem when you're eating a standard American diet. Good news is sweat eliminates these toxins, literally better than urine. They've done skin patch testing. It's like a nicotine patch without the nicotine. And they have groups of people that go in saunas and groups of people that don't go in saunas. And the ones that go in saunas, when they take off those patches and test for chemicals, they're full of BPA, phthalates, all kinds of, chemicals, toxins. Whereas the people that didn't go in the saunas, they don't have any of that on those patches. And what's crazy is neither group has a lot of these toxins in their urine when they do these studies. So basically you're not peeing them out, but you are sweating them out. So that's a key way to kind of accelerate some of the helping your body get rid of these toxins. Here's a picture of an actual isolation procedure from fat cells. You got a piece of fat. You literally put an enzyme in there to break it apart, to dissociate it, so then all the cells separate. And then you do some fancy centrifugation, some spins. You spin out the cells, and then you grow them in a plastic dish. Unfortunately, it's plastic, although you can use glass. And the reason I say unfortunately is because plastics do have estrogen chemicals. Not even if it's BPA free, there's a chemical called phthalate, and that's also estrogenic. It also acts like estrogen, but a different topic for a different day. So here's a picture from my own research, Zjuve light, and here's some cells growing in a dish. And this is infrared light, and I'm putting it on adipose-derived mesenchymal stem cells. That's AMSC. And what I do is I shine the light on these cells, and then I isolate the messenger RNA. You can use a kit for that. And, you know, basically you can revert that back to DNA using an enzyme called reverse transcriptase. It's a viral enzyme. So you take out the mRNA, you revert it back to DNA because DNA is super stable. And then you can basically measure how much of that gene is there, how much is expressed. And I do that for all 25,000 genes. So a little bit complicated, but you know, a little insight into what I do. And for me, the goal is to reprogram stem cells, right? Upgrade stem cells. Scientists call it preconditioning, and it's becoming a little bit bigger of a thing. I feel like when I first started doing it, nobody was doing it, nobody was talking about it. Now some people are doing it finally. And one of the chemicals they're testing is rapamycin. They're putting that on stem cells, and it's known to increase autophagy. Autophagy is basically cells recycling trash, cells taking out the trash, things like that. So a really beneficial process. And when you have these conditioned stem cells where you put rapamycin on them, then you inject those stem cells into animals, a lot more healing properties there. So a good example of, you know, how powerful this technique can be. And of course, rapamycin is a drug you probably don't want to take because it can harm your immune system. It can damage your immune system. At least in my opinion, I'd be really cautious with that. But again, if you're putting it on the stem cells and then you're injecting those stem cells, rock on, there's no issue. You're not actually taking in the rapamycin. They've done this same preconditioning protocol with low oxygen conditions, which I'll talk about. They find benefits there. They've also added some hydrogen sulfide, which is cardioprotective. They've done it with metformin, they've done it with retinol, they've done it with all kinds of cool things. And you know, the thing about it, some of the complications, of course, with stem cell injections in general, everyone's DNA is different. So you respond differently than the next person. So if I take stem cells out of you, I take stem cells out of somebody else, one of you may respond to infrared light differently or that preconditioning process differently. And then, of course, like I say, a lot of people have McDonald's stem cells, so they have all kinds of pre-programmed problems in their stem cells. Sex matters, right? If you take stem cells out of a male, you take stem cells out of a female, you're going to have different results. Similar with age. If you take stem cells from an 80-year-old person, and even if you're re-injecting them back into that same person, it's going to be different than if you take stem cells out of a 5-year-old person and put them back into that person. So there's a lot of variation with age. There's a lot of variation, period. Epigenetics, what you did with your past history, right? Like how well you've been sleeping throughout your life, how much stress you've had throughout your life. These are marks on top of the DNA that build up on your stem cells, and they can cause problems with, you know, with some of these stem cell procedures. And then of course the FDA is another complication, and I'll actually bring that back up in a minute. So umbilical stem cells, right? That's what Neil Reardon uses down in Panama. And, you know, there's about 7,000 studies using umbilical cord stem cells. Really popular, really effective. I'm a fan. And basically they're excellent cells because they're somewhat protected from toxins because the placenta oftentimes picks up a lot of these toxins and it doesn't allow them in. Doesn't mean they're fully protected. And that's an interesting aspect that most people aren't thinking about. When you're getting these umbilical cord stem cells, what was the mother eating? If the mother was on McDonald's for the entire pregnancy, and then you're taking out those umbilical stem cells, right? Versus somebody who's living a healthy lifestyle, eating healthy, sleeping, all that, take out those stem cells, you've got two completely different things. Again, the problem in science, everybody's lumping that all together and kind of saying it's the same thing. And then they're arguing about how much variability. They're saying, oh, stem cells are useless because it's so variable. You can't seem to get, you know, you can't get replications and repeatable results. Well, again, that's partly because of this issue here. S.A.D stem cells, right? So, you know, artificial estrogens, heavy metals, all these toxins, you know, you got to be really careful. Now, I'm going to talk for a minute about this parasite fallacy, and you'll see how it ties into stem cells in a minute. But there's a modern philosopher who basically said that human fetuses are parasites because they just take nutrients from the mother. And you see a picture here of some women that are promoting that. And so let's step back for a minute and talk about that, because again, these embryonic stem cells are really important to understand. Now there's a scientist, three scientists actually, that were awarded the Nobel Prize for discovering a protein called green fluorescent protein, GFP. One of them I've worked with, Roger Chen from California. Back when I was in Boston, you know, we've collaborated on some projects. And this Shimamura, he's actually from Boston University, where I did my PhD, and he's the one that really isolated the first GFP, the first protein. He got thousands of jellyfish and purified this protein that makes them fluoresce green, which is pretty cool in itself, right? And what's crazy with that protein, you can take embryos out of these animals, like mice, for example, or pigs, or literally any animal, and you can put that gene into those first few cells. And as the cells divide, every cell in the organism has that green fluorescent protein. It has that GFP. And so you can see this picture here with these mice, for example, 3 of them actually had the gene, and then 2 of them were unsuccessful. So sometimes when you put the gene in, you know, the cells spit it back out, it doesn't get in there, whatever. And with these pigs, you can see the same thing. Of course, they have some fur and stuff, so they don't fluoresce quite as well as these little baby mice, but it's pretty interesting. You know, you can turn a whole animal green with this protein. Now, it sounds really impractical and ridiculous, but here's the thing, right? There's a scientist named Hina Chundri, Harvard scientist, and she found that when you have injured mice, at least the initial study was with mice, and what she was doing was she was poking them with a needle, basically like giving them lung injuries by puncturing their lungs, for example. And these were pregnant mice. And what was really interesting about these mice, she was studying the healing, but again, they were pregnant, and she had taken the embryos and turned them green. So the mother mouse was totally normal, but the babies, the fetuses, were all green. They were these GFP mice. And what was super interesting, and again, we've known this since 2012, when you have these injuries to the lungs, to the heart, to all over the place, what happens is you have green fluorescent cells at the site of injury. So in other words, when the lung gets healed, you can open it up, do a dissection, you know, you can look at that site and it's full of green cells. So clearly not a parasite, right? Because there's obviously some cell transfer between the fetus out into the mother when you have injuries. Those are stem cells. And it's an example of how healing stem cells can be. And here's a picture of one of those green cells. And again, the mother has no green cells, so you wouldn't expect to see green cells except because they're coming from the fetus. What's interesting, of course, is they've done further follow-up research with that. And they find, like I say here, male fetal progenitor cells persist in maternal blood for as long as 27 years postpartum. And again, that's based on human DNA studies. So with humans, obviously, you're not turning people green and checking that out, but you can tell if it's a male versus a female cell. You can look at the DNA and tell if it, you know, determine if it's the fetus. And so we see that similar trend even in humans. And not only that, we see it in the brain. So there's a fascinating ability for these cells to go through the blood-brain barrier and actually heal the brain. So a lot of potential here. And the other aspect about parasites, so certainly parasites don't heal, you know, a mother that's injured or an organism that's injured, but they also don't do something called homing. And stem cells certainly do. Stem cells home on sites of inflammation. And so when you have, for example, mice that have a damaged pancreas from type 2 diabetes or things like that, if you infuse them or inject them with umbilical cord stem cells, you can do it in their leg and it'll find its way to the kidney, right, or the pancreas. And you don't really find those cells in other places. You predominantly find them in the damaged organ. So basically, when there's an organ that's damaged or a site of injury, it's secreting inflammatory chemicals. These stem cells are going around, oh, they found the chemicals, they home in on that site. They've also done this with x-rays to damage cells, to damage certain areas of mice. And so, you know, they basically have developed this hypothesis, and they've found a lot of different homing, a lot of evidence for this homing. Here's an image of that. And you can measure that with a lot of different ways. Like I say, you can make the cells green and look at the color. You can do it with all kinds of other interesting lab techniques I won't get into. And then the other aspect of stem cells, of course, is the exosomes. And so one of the things that they do, once they get to that site of inflammation, they secrete exosomes, which are a healing factor at that site of inflammation. And usually the stem cells themselves die, but the exosomes are there, and they've already reprogrammed those cells and assisted those inflamed cells, those damaged cells. And exosomes, of course, right now have gotten kind of a bad rap with the COVID thing, because a lot of people are talking about how viruses are just exosomes and they're conflating the two, which isn't true. Exosomes are secreted by your own cells, and they can be very healing in general. And again, stem cells are secreting exosomes. The thing about exosomes, and you can see the image here, it's just one cell, pinching off a piece of membrane, sending it out, picked up by another cell, and that cell has an impact from it. So they're externally secreted bodies. They contain DNA, RNA, proteins, of course, fats, because they have a little membrane on them, just like your cells do. And they're communicators, so the thing is a lot of scientists are super excited about these, because just the fact that we have RNA in those exosomes indicates they can go to other cells, dump that RNA in, and then immediately have an impact on cells. And it can be long term. It can, they can do your cells can communicate, from your toes all the way up to your head using these exosomes. The reason a lot of scientists like them, of course, is they're super simple and they can be designed. And this is an issue, they can be patented, right? So you can do a clinical study just with one exosome, and you've got a patent on that. You do the whole multi-year clinical trial, $1 billion on average at least, and you've kind of cornered the market with that exosome. So this, to be honest, is why most people are so excited about exosomes as opposed to stem cells. And there's been a lot of pressure to basically say, yeah, stem cells aren't that big of a deal, but exosomes are awesome. And yes, they are, but it's a little more complicated, right? And the reason everybody's doing, a lot of the field at least is shifting in that direction is because, there's money in exosomes. So let's see. So basically, the problem with stem cells, contrasting, stem cells can't really be patented. Your body makes stem cells. There's no potential for patenting that. But the beauty of stem cells is when they get in there and they home in on those sites of inflammation, they know what to secrete, right? Again, I said there's millions of different ways, maybe thousands, maybe tens of thousands, millions of different things these cells can secrete in exosome packages, right? Proteins, mRNA, DNA, there's all kinds of stuff. And the stem cells, when they find a site of inflammation, they know what to secrete. The problem with patenting one single exosome and injecting that, who knows, right? Who knows if it's the right one for you, the right one for your age, the right one for your sex, the right one for your diet, all that. Again, the stem cells are a lot smarter than we are in that regard, because they're in there, they're on the front lines, and they can secrete millions of different options. So that's really why I'm a bigger fan of stem cells than exosomes. But I see the purpose of exosomes. They're potentially great, but again, they're more limited. And, again, people are really pushing these exosomes. So what's the FDA stance? In 2017, the FDA cracked down on stem cells, and they only allow minimal manipulation. That's the term. And what that means is you can't take stem cells out of your body, like from your fat cells, grow up a ton of them, and then put them back in your body. That's against this minimal manipulation law. So what you can do is you can take them out of your body and then put them back in your body. Well, they were already in there to begin with. I don't see that that's that big of a benefit. So there's been a lot of push in the field has been trying to get the whole industry towards exosomes and to move it away from stem cells. And I mean, it makes sense, right? Because there have been actual fraudulent issues with people marketing stem cells that are garbage. I mean, there's certainly some validity here, but there's all it's a big can of worms. But you know, I work with these orthopedic surgeons, and certainly they would rather do a full knee replacement or a full hip replacement. At least the hospital would rather have them do that than just a simple injection of stem cells. And again, if they can get a patent on the exosomes or something, well, then there's more money there, so they want to pursue that. So usually it's just a follow the money issue. So because of these restrictions, of course, a lot of people, a lot of optimizers, a lot of bodybuilders I know, for example, they go overseas, they get these stem cell injections. Neil Reardon, like I mentioned before, has had great success. He charges outrageous amounts of money though. So not everybody can afford 25 grand for some stem cell injections. But I also know people down in Colombia where they charge closer to $1,000. And I know people personally that have been there with success. And then, you know, I'm always interested in your opinions and what you have found with your research in terms of injecting stem cells, because there's certainly a lot of variation. There's some clinics that are amazing, some are terrible, some are injecting dead stem cells, right? Which still there's some validity even there, because those stem cells have already secreted a bunch of exosomes. So at the end of the day, you're just doing exosomes and under the guise of doing stem cells. But you preferably you want to inject actual live stem cells and let them figure out what your body needs, where it needs it. So why would you inject stem cells? Well, stem cell factor, SCF, it declines with age. It's a protein secreted by stem cells. And low SCF is associated with all kinds of health issues, declining organ function, for example, Alzheimer's disease, for example. So you don't want low SCF. You don't want low stem cell factor. But again, as you age, it decreases. Not only does the stem cell factor decrease with age, their potential decreases. So their ability to divide and replicate decreases. So they kind of get sluggish. They get kind of old, the stem cells themselves. The actual numbers decline. So here's a graph showing, you know, like per cell in your body. When you're newborn, 1 out of every 10,000 cells is a stem cell. But then even as you're a teenager, it's 1 out of 100,000 cells, right? And then as you age, you can see it declines and declines. And by age 80, it's 1 out of every 2 million cells in your body is a stem cell. So there's this huge ratio imbalance where you hardly have any stem cells. You get a lot of inflammation. Your body can't react to it. It can't do much to it. So you want to bump it up. There's another factor called LL37 that stem cells secrete. And basically, younger stem cells produce more of that. It's an antibiotic, you know. So a lot of things going on. I mean, let's talk natural though. Let's switch it over. We've talked about injecting stem cells. What can we do to naturally upgrade our own stem cells, our S.A.D stem cells? One area that's been studied that's interesting is metformin. You can actually precondition stem cells. You put the metformin in with the stem cells and then you re-inject them. And of course, metformin has been studied because it lowers blood sugar, it improves metabolism. And like I mentioned here, it rejuvenates aged stem cells. The thing about it, and I kind of say quote unquote metformin is a drug because in some countries it's not even prescription, but in America we make it a prescription. It's derived from a French lilac plant. It's pretty natural. But what's way more natural than metformin? Not eating standard American food for starters, but also just lowering your blood sugar, increasing your metabolism with exercise, with good sleep, and even better with ketosis, right? You're certainly going to lower your blood sugar with ketosis, the keto diet. So that's a lot more natural than metformin. Has anybody done that study? Not that I know of. But they have done studies with fasting. And of course, fasting imitates ketosis as well, because you have a lot more autophagy, and we'll talk about mitochondria later. But in 2018, MIT researchers found 24-hour fasting increases gut stem cell size and improves the stem cell function. So you don't have to inject stem cells to improve that potential, right? That stem cell potential that's declining with age. You know, you can improve it just by fasting. And I think literally everybody should be fasting. You know, I do DNA analysis and everybody has genes that indicate they should be doing some kind of fasting. And maybe not literally, there's always the exception. There's always somebody out there who responds pretty poor to it. But again, it's pretty rare. So hormone balance, that's another big factor with stem cells. So here's that stem cell potential image again. Stem cell potential declining with age. In this particular study, which again, references, if you want them, I can get them to you. I didn't want to clutter up all the slides, but stem cell potential has been hypothesized and studied with testosterone, with steroid hormones, because that can reverse a lot of that. That can reverse some of that declining stem cell potential and bring back the stem cells into a younger looking stem cell. They call it a phenotype. And estrogen certainly has been known to increase stem cell numbers, which is very cool because you actually upregulate the amount of stem cells you have, not just the stem cell potential. Testosterone has not been shown, does not increase the numbers of stem cells, but it does, of course, if you have higher levels of testosterone, you also have higher levels of natural estrogen, which is good. Natural estrogen, totally different than artificial. Artificial BPA, phthalates, things like that, really problematic. The natural estrogen can be really beneficial. It's protective for your heart, protective for your brain, protective for your arteries, a lot of things. But the ratio really matters. If your testosterone is super low and your estrogen is super high, that's a huge problem. If your testosterone is high and your estrogen is high, that's usually really beneficial. And your testosterone naturally gets converted to estrogen. So one of the ways to bring up your estrogen is to bring up your testosterone. So again, even though testosterone doesn't necessarily increase the numbers of stem cells, it does increase stem cell function. So both of these sex hormones, super beneficial for your stem cells. And, you could almost give an entire talk just on how to increase testosterone, how to improve your sex hormones. But certainly one of the aspects of it that I always talk about is BPA, because BPA and artificial estrogen, not only does it decrease your total testosterone, it decreases your free testosterone, it mimics estrogen, it blocks binding to the testosterone receptor. So it's basically desensitizing your testosterone. So, you've heard of insulin sensitivity, for example, and how a lot of Americans have really terrible insulin sensitivity. Well, they also have really terrible testosterone sensitivity. So even if your levels of testosterone look really good on a blood test, a lot of people, because they have all these estrogen chemicals blocking the binding to the receptor, they're basically having no response from the testosterone. So it's like testosterone desensitivity, or a better word would probably be testosterone resistance, just like people say insulin resistance. You definitely don't want that in terms of your stem cells and pretty much anything else relating to health. Another way to naturally upgrade your stem cells, your standard American stem cells, if you have them, infrared and sunshine. As you know, I directly do research with infrared, but sunshine, right? I mean, 50% of the sunlight is infrared, and only 5% of it is about 5% is UV. And studies kind of indicate that infrared is beneficial by looking at wound healing with these AMSCs that I look at. And of course, I have my own research that's not published, that I'm not going to talk about, but infrared is definitely upgrading these stem cells. The other thing about sunshine that's really interesting is people think of it just in terms of vitamin D, and that's awesome. And that's probably my next bullet, is that vitamin D, of course, improves stem cell function. But people don't think about melanotan, which is an interesting peptide that your body makes when you have exposure to sunlight. And melanotan acts on this MC4 receptor, this melanocortin receptor, and that decreases hunger for one, so it helps you regulate your appetite. And it's beneficial for stem cells as well from that perspective. So a lot going on with sunshine, super easy, a lot cheaper than going to Mexico and going to Colombia and injecting stem cells. Retinol is another one that's massively important. And I didn't say keratin, I said retinol. Talk about that in a second. It supports the self-renewal of stem cells. So again, stem cell potential is declining with age. Help to reverse that by taking some retinol. The problem with retinol is most people think they're getting retinol, but because it's called vitamin A, but so is keratin. Keratin is also vitamin A. Now, keratin is in plants. So if you eat carrots, people say, oh, eat carrots, they're good for your eyes. Right? It's because they have a lot of keratin. They're orange. Keratin is, literally, that's the hallmark of keratin. It's orange color. The problem is a lot of people, they don't convert keratin very well to retinol, and your body doesn't use, your eyes don't have keratin. They have retinol. You have to convert the keratin to retinol. And about 60%, there's people out there with this genetic issue that I see pretty regular. It's on the BCO1 gene. And I give you the code here for that gene if you want to look up your version with some software like Promethease. But a lot of people have literally a 60% reduced conversion of keratin to retinol. So you're not going to get enough retinol by just eating plants. That's the point. You know, you have to eat liver to get retinol. You have to eat egg yolks or butter, although liver is by far the best source. So I recommend grass-fed liver. If you're not sure, if you haven't done your DNA, it's tricky because liver also has a lot of iron, and some people have really terrible iron genes, and you don't want to get your iron super high, or that can damage your arteries. So in that case, you want to go with actual retinol supplements. And cod liver oil is a good example of something like that. Again, animal products, retinol, plants are all carotenes. And those carotenes aren't going to help your stem cells if your body's not converting it to retinol. Oxytocin is another one that naturally upgrades your stem cells, and you don't have to inject it, although I know people that do. There's an article here by Self-Hacked. I'm a scientific advisor for that website, and I love it. And they have 34 ways to naturally increase your oxytocin. It's considered the bonding hormone. Relationships, good relationships with people, super important. Dark chocolate, another easy way to increase your oxytocin. 34 ways, right? There's a lot of ways. And that, again, it increases stem cell numbers and stem cell function. So it's a win-win with oxytocin. Super interesting here. I gave this talk in Texas before this COVID thing, and or a version of this talk. And I showed this slide, and I had a lot of people that wanted the reference, and it's a great reference. It's very clear. They'd done a study with literally just one single 2-hour hyperbaric oxygen treatment, and they found that doubles stem cell numbers. And, you know, and this is people, this is not mice, right? And not only that, they had 20 treatments that increased, you know, if they did 20 hyperbaric treatments, it gave 8 times higher stem cell numbers, which is insane. So, you know, some people inject stem cells and barely get a bump in how much stem cells they have. They don't get 8 times more in most cases, right? And again, that's complicated because most of the time when these clinics are injecting stem cells, they're pretty much McDonald's stem cells. So who knows if they're even that good. But assuming you've got awesome stem cells, as you age, hyperbaric can be really beneficial to bumping up your natural levels. It's a little bit more involved, of course, than just going out in the sun or something like that, or getting retinol. But again, it's cheaper than going to Mexico. Many of the preconditioning studies, you know, again, they're manipulating oxygen. They've also found that people exposed to altitude have a lot of benefits to their stem cells. So that's another way to manipulate your oxygen levels. And speaking of oxygen, mitochondria, this is the last thing here. You know, what's cool about mitochondria, of course, stem cells have mitochondria, right? It's the battery power of the cells. It makes ATP, energy. But stem cells have this crazy thing they can do. They can literally squirt out their mitochondria. They can transfer their mitochondria into other cells. And this has kind of been researched by these 2 doctors, McCulley and Emeny. I don't know them, but they're at Harvard, and they've had good success literally just taking out mitochondria from cells and injecting them, particularly with organ failure. And I think it was, yeah, here's the New York Times article. Again, that's an option. You know, you'd maybe do that in a really extreme case, but what are some of the ways to upgrade your mitochondria naturally? Well, fats, right? Eating healthy fats, teaching your body to burn healthy fats. And there's a huge metabolic difference between sugar and fat in relation to the mitochondria. There's a book called Tripping Over the Truth, where Travis Christofferson, I think his name is, the author, really goes through this really well, in particular in terms of cancer, because cancer cells have this weird ability to burn sugar. It's called the Warburg effect, W-A-R-B-U-R-G. The guy Otto Warburg got a Nobel Prize for discovering it. Basically, cancer cells burn sugar. It's like the hallmark of, everybody knows this. We don't know exactly why, whatever, but cancer cells love sugar. And one of the crazy things about sugar, and you can see this image here, kind of says, oh, here's sugars and fats in cells, and then they go into the mitochondria and get burned for energy. This picture is pretty classic in a lot of beginner textbooks and things. It's not really true. It doesn't really work that way. What actually happens with sugar is your body can break sugar apart outside of the mitochondria. So it comes into a cell and it breaks apart and makes a couple of ATPs and lactic acid. So you're getting some energy from sugar. It's really fast, but it's not even using the mitochondria. Now, what's crazy about fats only can be burned in the mitochondria. They can't get split out and make lactic acid and all that kind of stuff outside of the mitochondria. They are 100% of their metabolic process is inside the mitochondria. Sugars have that option. They can you can make a little bit of ATP. You can make like 6 ATPs or something, or 3, I can't remember, outside of the mitochondria. And then you can also transfer sugar into the mitochondria, and then you make about 36 ATPs. So sugar, 36 ATPs if you actually use the mitochondria, but your body has the option. The cells have the option. With fats, no option. They only go into the mitochondria, and when they do, and they get burned, they make over 100 ATPs. So far more efficient, in terms of energy, fuel source, but also far more mitochondria utilization. So if you're eating sugar all the time, your mitochondria are just getting fat and lazy, they're shutting down. You eat fats, you're forcing those mitochondria to burn them. So anyways, natural versus injections. I mean, I'm a big fan of just naturally upgrading your stem cells, of course, all the basics, do keto, do exercise, do intermittent fasting. Hormones are really important to balance, sunshine, retinol, like I say, relationships with that oxytocin, all the fundamentals, right? The injections can be particularly valuable if you've got some extreme injury, or if you really struggle, with inflammation. And again, it's really variable though, because those injections, you got to be careful where you're getting those stem cells. A lot of mothers these days, they're eating McDonald's, their stem cells are McDonald's stem cells. You're not doing much good for yourself. You're spending a lot of extra money injecting other people's McDonald's cells into your body. So yeah, and then of course, age is probably the most important aspect or the best argument for injecting stem cells, if you're really going on the extreme to optimize your health. But like I say, hyperbaric is probably that next level up too. So thanks. That's the stem cell talk. I look forward to questions on my email. You can find me at the AJ Consulting Company website there. So take a screenshot. And like I say, I have my email on there. Shoot me a question. I appreciate you hanging out with me.